In this video, we are going to learn how to make gene ontology or Go annotations using Canto. A Go annotation links a gene product, so an RNA or a protein, to its normal or wild-type function and cellular location, as well as to the processes it is involved in. There are three types of Go annotations. Molecular functions, which describe the basic single-step activities of gene products. Some examples are microtubule binding and protein serine threonine kinase activity. Then we have biological process annotations that describe a series of events accomplished by one or more ordered assemblies of molecular functions. Some examples are the mitotic spindle assembly and the NADH regeneration. The biological process ontology also contains phases, such as the mitotic M phase. This cannot be used to directly annotate a gene product, but they can be used in annotation extensions, as you will see later. Finally, we have cellular component annotations that indicate the subcellular structures and macromolecular complexes where a gene product may be present. Some examples are the nucleus and the proteasome complex. To better understand Go annotations, let's look at the role of CDC15 during mitosis. CDC15 goes to the medial cortex during mitosis, where it recruits other proteins such as CDC12, and the recruitment of these proteins is important for the formation of the actomyosin ring. Now, let's find the elements of this model that are relevant for Go annotations. First, we have two gene products, the proteins CDC15 and CDC12. We've mentioned that CDC15 is recruited to the medial cortex, which is a cellular component, and that it recruits CDC12. This is a molecular function, cytoskeleton protein membrane anchor activity. Finally, this activity is involved in the biological process mitotic actomyosin contractile ring assembly. All this occurs during mitotic M phase, which is a special type of biological process. We can capture part of this model by making a Go annotation saying that CDC15 has the molecular function cytoskeleton protein membrane anchor activity. We can provide further details through annotation extensions that link the molecular function to other entities. For instance, this molecular function happens during the phase mitotic M phase, and it occurs in the cellular component medial cortex. It also is involved in the biological process mitotic actomyosin contractile ring assembly. Certain molecular functions have special extensions. In this case, we can say that the molecular function cytoskeletal protein membrane anchor activity of CDC15 anchors CDC12. In the same way, we can make a biological process annotation. We can indicate that this biological process happens during mitotic M phase and that it occurs in the medial cortex. Finally, for a cellular component annotation, we can indicate that the gene product is observed in this location during mitotic M phase. Go annotations also support extensions to indicate the active forms. For example, we can say that the function, the process, or the presence of a gene product at a cellular component only happens when it is phosphorylated or when it is palmitoylated. For now, this is hard to annotate in Canto, so just add a comment indicating this in the annotation. To make a Go annotation, you must have added the involved genes to the session first. If you haven't, click on Edit Gene List. If you don't know how to do it, see the link in the description. To create a Go molecular function annotation, click on the quick links, select the gene you want to annotate to, and then look for the Go term that better fits your need. If you don't know exactly what term to use, look for a broad term and then narrow down with the search. We're going to write protein and membrane, and we find protein membrane adapter activity. We click on it, and we always verify that the definition matches what we expect from the term. Then we click on Next, and we see whether we can find a more specific term, in this case, cytoskeletal protein membrane anchor activity. We click on it, and if we're satisfied with the specificity of the term, we verify the definition, and we click on Finish. If you can't find the Go term you need, click on Suggest a new term and suggest a name and a definition. We can choose the evidence code. So what is the experiment that supports this Go annotation? In this case, let's say it would have come from an experiment, and then it's good practice to indicate the figure or table where this comes from. If you know that a certain modified version of the protein is the one that presents this molecular function, you can also indicate it in this box. So in this case, we can say that is the unphosphorylated version. Finally, we can use the annotation extension to link this annotation to other entities. For example, we can say that it anchors the protein CDC12. We can also indicate that this CDC15 function is involved in a biological process, in this case, contractile ring assembly. Like before, we can use Next to find more specific terms. For example, mitotic actomyosin contractile ring assembly. Once we are satisfied with the term, we verify that the definition matches our expectation, and we click Finish. We can also indicate when this CDC15 function takes place. And this can be a cellular response to stress or a cell cycle phase. So for example, it could be cellular response to heat, or in this case, mitotic M phase. We pick this, click Next, Finish. We can also indicate the physical location where this activity takes place. In this case, it is the medial cortex. As before, we verify the definition, we click on Next, we try to find a more specific term. If we don't, we click Finish, and we're done with this annotation. Once we have completed all these fields and we've added all the annotation extensions we can click on OK. Making Go biological process and cellular components annotations is basically the same. Just click on the right link here and follow the same steps as before. Again, always click on Add and see if you can provide annotation extensions. For more information on annotation extensions, visit the link in the video description. Once we have created a Go annotation, Canto has tools to speed up the process for future ones. For example, we have here that ACE1 goes to the mitotic spindle midzone during mitotic anaphase B. We can click on Transfer and make the same annotation for other genes, for example, PEC1, KLP9, and ARC1. We can decide or not to transfer the extensions by clicking here. We have then transferred the annotation to these other three genes. Another useful tool is the Copy and Edit button. Say that we want to add another protein that CDC15 anchors apart from CDC12. We can click on Copy and Edit, edit the extensions, remove CDC12, and add another anchor, for example, ETD1. This creates a new annotation with has input ETD1 instead of has input CDC12.